All right, then let's get started. So first of all, during this difficult global pandemic time, I'm very glad everybody can join us, and hope everybody is safe and stays safe. So as Jacob just mentioned, I'm the first speaker for this summer course, and hopefully I can give you a general view of the neuroscience and give you a sense of how neuroscience is performed, what we are interested in, and also for the title you can see we are talking about the cells or the cell types in the nervous system. First of all, I would like to give you a brief introduction about myself. My name is Yang, or Yang Zhang for the full name. Currently, I'm a principal investigator at the Neuroscience Research Institute at Peking University in Beijing. And also I have a second appointment with the IDG McGovern Institute for Brain Research at Peking University. So in terms of my research experience, actually, I went to the United States first in 2001 as a visiting scholar. Started my research career at UT Southwestern Medical Center at Dallas. And then I joined the BCMB PhD program at the Johns Hopkins University School of Medicine. There I got my PhD in 2008. Actually, I have a very diverse research background. For my PhD, I was not in neuroscience at all. I was studying cancer biology and tumor research. But I have been interested in neuroscience since very early days. And after my PhD, I decided to join Rick Huganier lab to study neuroscience. And then I stayed there from 2008 and 2016 as a postdoc fellow, and then came back to China in September of 2016 and established my own lab. So what my research focus is called a phenomena called synaptic plasticity. Of course, this term might be novel to you now. Later on in the course, I'll introduce you in a great detail. So synaptic plasticity is a phenomena that plays a critical role for learning and memory. So you might remember or recall an experience like 5 years ago or 10 years ago, right? That's because that event was encoded in the memory. You are able to recall. So this is a very interesting phenomena in neuroscience. So in terms of synaptic plasticity and physiological conditions, it's essential for learning and memory. In the meantime, if something goes wrong for synaptic plasticity, then a variety of neurological or psychiatric disorders will appear. So in terms for my lab, so we are focusing on to elucidate a mechanism underlying synaptic plasticity and learning and memory. So after the brief introduction about myself, next we'll give you the outline of this lecture. So first, we'll start give you a brief introduction of the central nervous system. And then we'll move on to a little bit in detail to talking about synaptic transmission and synaptic plasticity. And in the end, I'll introduce you a little bit about the research I have done in my lab. So the major goals in neuroscience actually are kind of twofold by my definition at least. First of all is understanding how the brain works. As we all know, the brain is a very complex organ. It literally controls almost every behavior of our daily lives. So the mystery of the brain has been the interest of neuroscientists for a long time, as we'll talk a little bit more in detail in the following slides. So understanding how the brain works is one of the major goals in neuroscience. Of course, if anything goes wrong for the normal brain, then we'll have a lot of neurological or psychiatric diseases. So the second major goal in neuroscience actually is to discover new ways to prevent or treat brain disorders. So as I just mentioned, the human brain is a very complex network. By estimation, it has over 100 billion neurons in the human brain. So on the left, it shows this diagram illustrates how the human brain looks like. As we can see, we have the cerebral cortex, we have the cerebellum, and something in the middle. In the middle, there's also subcortical structure in the midbrain, and also we have hypothalamus, we have the hippocampus, we have the basal ganglia, and we have, a bunch of structures in and between that construct the complex organ of the human brain. So most neuroscientists will agree that the human brain is the most complex organ in the universe. By the number, you can see it has over 100 billion neurons. And on the right, it shows the dissociated neuronal culture in a petri dish. You can see all the neurons are not isolated, right? They are interconnected with each other so that they can communicate with each other and also transfer information between different neurons. So the study of neuroscience, or the study of the functional brain, actually began more than 100 years ago. One figure I want you guys to remember is a Spanish neuroscientist, Santiago Ramón y Cajal. He is widely regarded as the father of modern neuroscience. So back then, he uses just very simple equipment, actually, the light microscope. He was interested in the structure of the brain, actually trying to understand what consists of the brain and how many. Back then, it's not well defined. Now we know it's called how many types of neurons are there in the brain. So on the right, that's one of the hand drawing of Cajal, actually, after his experiment. We'll talk a little bit more in detail later on too. So for Cajal, what he did is he utilized a method called gauge staining, so which can randomly outline a sparse population of neurons in the brain, and then he can observe under this light microscope the morphology of those neurons. So after a series of gauge staining, he observed a variety of neurons in the brain, and based on his understanding and on his insight, he drew the map of the brain on the right. So basically, we can see this is a rough structure of the human brain by his recognition back then. I'm sorry, there's someone else talking and have a little bit distraction. I thought he has a question. So on the right, we're talking about the neurons he observed in his gauge staining. And you can see there's a variety of neurons with different shapes, and they have a different location. And also, amazingly, if you look carefully, there are some arrows he draw on the map too, which indicate, based on his understanding, that's the information flow. In another word, he also thinks that all the neurons are connected. They are not they are not in isolation. They're connected with each other, and they pass information from one neuron to the next. Of course, we call him a genius, or the founding father of the modern neuroscience, for a reason, because he really provides some insight for a neuroscience structure. Even as of today, when we look at back his drawing, his understanding of the neuron, of the information flow, it still holds truth as of today. Because of his outstanding contribution, he was awarded the Nobel Prize in 1906 together with Golgi. So as we are saying that the nerve system is consists of a lot of neurons. Other than neurons, there's also glial cells. Actually, based on today's understanding, the nerve system consists of neurons and glia. In the diagram A, give you a cartoon picture of how a neuron looks like. Actually, this is a typical pyramidal neuron. You see the cell body is, look like a pyramid, right? It's a triangle shape, if you like. And then it has a very beautiful dendritic processes extending apically. And also highlighted in red is the axon. So the neuron, those are, you can consider those three compartments as the essence for a neuron. You had an axon, has a dendrite, has a cell body. Of course, you can see on the dendrite, there are also a lot of protrusions. Those protrusions are called dendritic spines. We'll talk those structures later on in the lecture in great detail. And also, other than neurons, there's also another cell type called glial cells. It also play an important function in the central nervous system. So you might get the idea from Cahall's drawing that the neurons are not identical. Every neuron has its own shape, might have its own function. So there is a diversity of neurons in the central nervous system. Again, here is a cartoon showing the neuronal diversity. So on the left, there is the spiny neurons or excitatory neurons, as you would call it. On the right is the non-spiny inhibitory interneurons. So other than morphology, what else can we categorize?
Dr. Zhang, are you able to hear us?
Yes.
Sorry, your internet went out for a minute. Would you be able to go back to the slide where you were talking about spiny and aspiny neurons and go from there?
You mean right here?
Yes.
Okay, I'm sorry. So
it's okay, thank you.
With the internet. Well, what we are talking about, there's a neuronal diversity. There's a lot of type of neurons in the central nervous system. So this is a simple categorization based on their morphology. We can see there's spiny neurons on the left, and there's non-spiny neurons on the right. And also all the spiny neurons are typically the excitatory neurons and also the pyramidal neurons. As we are seeing, we got like 6 layers in the cerebral cortex, and for each and a different layer, they have a different type or the different type of spiny or projection neurons in each layer. As outlined here, the black indicates the cell body and the dendrite. The red is the axon. And also on the right is the inhibitory neurons. As you can see, morphologically, they are different. And also another characteristics for the inhibitory neurons is they don't have spines. That's why they're called non-spiny neurons. So for the inhibitory neurons, similar to the excitatory neurons, it also allocated in the different layers of the cerebral cortex, and they can be further categorized into different type of inhibitory neurons. There's basket neurons, chandelier neurons, bipolar, railing neurons, and such. And those inhibitory neurons also play important role, modulate the activity of the excitatory neurons. So you might wonder by now, how many type of neurons are there in the brain or in the central nervous system? That's a great question. Depends on how are you going to categorize those neurons, or what's your criteria for categorizing those neurons. So simply based on morphology can be categorized into those subcategories. One is unipolar neuron, the other one is bipolar neuron, and also there's multipolar neuron, and also pseudo-unipolar neuron. So those are the simple cartoons give you a sense of the morphological differences between different type of neurons. So other than morphology, or in another word, if neurons appear in the same morphology, are they exactly the same type neurons? The answer is no. So even if they appear the same in morphology, they might carry a different function. They might have a different physiology. So here, this diagram shows the neuronal types in the retina, actually. So you see the retina is a very complex organ, if you would call it, and it senses the light coming in and form the signal, and then relay the signal to the visual cortex. We can see we have rod and cones. Those are the photoreceptor cells. And then we have horizontal cells. We have bipolar cells, amacrine cells, and then we have retinal ganglia cells. So for those cells, they can have exactly the same morphology, but their physiology can be different. There are some light on cells. There are some light off cells with exactly the same morphology. So in another word, even if neurons, they look like the same, they might function different. So those can be categorized into different type of neurons. Even if they have the same morphology, they have the same function, at least the general function. And in terms of their detailed, let's say, protein expression or gene expression, it also could be different. So nowadays, there's a technique developed called single cell sequencing, have been identified more cell types in the central nervous system. So the gene profiling is another way to categorize neuronal types. So by estimation, actually, there are up to 10,000 type of neurons in the central nervous system. Depends on how detailed characterization you want to go. So next, you might wonder, so where are those neurons come from? Where are all these type of neurons come from? This process is called neurogenesis, actually. Of course, we all know all the human body come from a fertilized egg. And the brain or the central nervous system is part of the fertilized egg. So where it comes from, it comes the neural stem cells or the neuronal progenitor cells. So this diagram shows where the neurons or how the neurons develop into mature neurons. So of course, start at the embryonic stage, continue to develop into the early postnatal stage, and also throughout the adult life. So the neuronal burst, or it started with a single cell or with no processes, and then it went through migration, it went through morphogenesis, and then it went through synapse formation, and then at the end, it has a modification of the synapse synaptic connection. So what it means is, as I said in the earlier introduction slide, so all the neurons are connected. So they are not in isolation. So they are connected at a specific structure, which is called the synapses. And later on, we'll talk into a little bit more detail about the synapse formation. So here, this cartoon give you a brief idea about how a neuron can start from a single, like a round shape, not much neuronal processes, a cell develop into a fully developed neuron with a complex dendrite, a lot of spines, and also have the axons to communicate with other cells. So by definition, the neurogenesis is a process by which neurons are generated from neural stem cells and neural progenitor cells. And in general, there are three major steps in the neurogenesis. One is cell proliferation. The second one is cell migration. And the third one is cell differentiation. It happens in a sequential way in somewhat, but also is intermingled. So first is an overview of the projection neuron generation in the mouse cortex. Again, the timeline is at the bottom, right? We start from embryonic, less than embryonic day 10 is on the left, and then E11, E12, E13, 14, 15, 16, and then 17. So in terms of neurons, it all started with the ventricular zone. That's a specific structure at certain step of the development that the neuronal progenitor cells are generated. That's why on the left corner it's called MP. It stands for neuronal progenitor cells. Then it went through a proliferation, and then through migration, and then the differentiation and into different layers of the cerebral cortex, some in the layer 6, some in the layer 4, some in the layer 1 and 2. Of course, there's a certain patterning due to the time limit. If people are interested, you can go look at the literature. I'm not going to talk too much on those development of the different layers of the cortex for this lecture. So as I said, there are 3 major steps in the neurogenesis. The first one is cell proliferation. So that's easy to understand, right? So we all started from a fertilized egg, and then the cells proliferate, and we have the inner layer of the cell, we have the outer layer of the cell, and depend on the positioning, and also depends on all the morphogenic factors, the location can induce the cells or different part of the embryo develop into different part of the human body later on. Of course, all this process need cell proliferation, or need the cells to grow, right? You have to grow bigger from 1 to 2 to 4 to 8, and that's how it works. So the cell proliferation in terms of neurogenesis, and there is 2 type of cutting, or 2 type of cleavage for the cell proliferation for the neural progenitor cells in another way. So one is called vertical cleavage. The other one is called horizontal cleavage. So for the last slide, I mentioned a specific structure is called a ventricular zone. So this is the ventricular zone. Again, it's at the bottom. So what happens for the vertical cleavage is the cells divide into 2, and it was cut vertically, divided into 2 cells. For this specific cleavage, both daughter cells stay in the ventricular zone. They can divide again and again. Again, they serve as 2 neural progenitor cells. In another way, or in another word, this mode happens earlier in the development. So this is trying to grow to get more cells, to get more neural progenitor cells for the later development. And also, this is in the stage to grow the critical mass for the embryo. So another type of cleavage is called horizontal cleavage, as seen in the bottom of the left panel. So the neurons are cut horizontally. So in this way, one cell left the ventricular zone, another one stays in there. So in a way, the cell left the ventricular zone, stop dividing. It's called the post-mitotic cell. Those are the neurons. They'll migrate to the place they're supposed to be, but they're not going to divide again. So for the daughter cell that stays in the ventriculum, it retains the capacity to divide again and to produce more cells for the central nervous system. So once the cells left the ventricular zone, it will start a process called migration. So then the cells will migrate to whatever place they're supposed to go. So here is a kind of a simple labeling experiment that were performed in the mouse cortex. So the cells were labeled with a dye called BRDU at a particular place, at a particular stage of the development. And then it was traced or sacrificed the animal at different postnatal age, and to see where those cells are labeled at 11 end up. We can see This is from E13, E15, E17. We can see the cells are migrating out, out again. So this is the cell migration pattern. And the panel B at the bottom of this slide is showing a cartoon how those cells migrate. In a way, I'll talk a little bit about the general pattern of different layers of this cerebral cortex. So actually, it's called an inside-out pattern. What does it mean? So it's the layer 6 neurons are formed first, and then layer 5, then layer 4, then layer 2, then layer 1. So there's a pattern, it's called inside out. So the neuroprogenitor cells produce the neurons in the layer 6 first, and then going outwards. And then, I mean, as I said, when the cells reach their destination, the cell differentiation part will start, or it's kind of somewhat intermingled. It does not have a clear cut line between the cell migration and the cell differentiation. So here is an idea or the illustration for the cell differentiation. As you can see, those are the neurons isolated from the embryos and cultured in vitro. So it's called, at the bottom of the screen, it's called days in culture. It's how many days those neurons are grow in culture. So first, once they are plated, all these neurons, they don't have too much neuronal processes. It's called lamellipodia. It's like those small protrusions and not very far away from the cell body. And as time goes on, as we grow those cells in the culture system, we'll have those minor processes and also the axonal outgrowths and the dendritic outgrowths, and then have the neuronal maturation. This is basically give you the idea of the timeline of the neurons starting from a single roundish cell, how to grow into a mature neuron with multiple processes for the dendrite, for the axon. As we have mentioned earlier in the presentation, the neurons in the central nervous system are not in isolation, right? They are interconnected. They communicate with each other. And also the structure neurons communicate with each other is called synapses. So here I'm showing you the ultimate structure of the synapses. Actually, those are the EM pictures of the synapses. So in general, there are two types of synapses in the central nervous system. One is called chemical synapses. The other one is called electrical synapses. So on the left is a chemical synapses. On the right is electrical synapses. So the electrical synapses is relatively abundant early on in the development, but as time goes on, as development goes on, and the majority of the synapses in the central nervous system or in the brain are chemical synapses. So we'll focus a little bit more on the chemical synapses later on in the presentation. So you can see from the picture on the left that in the upper, it's not a cell, the upper round structure is axon, right? The lower round structure it's called the spine, right? So the in the axon, you might say there's a lot of vesicle, a lot of bubble-ish like structure. So we'll talk this in great detail. Those are those called synaptic vesicles carrying the neurotransmitter. So this is the criteria for to identify a synapse or a chemical synapse in the EM structure. First of all, it has a presynaptic vesicle. This is this bubble-ish like structure, which is seen previously on the slide. Those called the synaptic vesicles, and also have an active zone or a synaptic cleft. Then it has a postsynaptic density. Again, we'll talk a little bit more details into that. So those are the criteria to identify a synapse on the EM picture. As we are seeing, those synapses, we have a presynaptic terminal, right? So what are those presynaptic terminals, right? So those the presynaptic terminals actually are the axons from the neuron in the circuitry, or the neurons projecting to the postsynaptic neuron. So here are a picture of the growth cone of the axon. So the growth cones are labeled in green in a way. So on the left, it's called it's the philopodia, and on the right is the lamellipodia. So in order for the synapse to form, so the axons will go out and search for its postsynaptic partner, or the dendritic spine. So this process called axon guidance. So the axons does not go out with no purpose at all, just randomly go out. It has some kind of a path, or has some kind of a destination that is guided, or the axons are guided to its destination, or where it's supposed to be. So this diagram shows the process of the axon guidance. So basically, it's a dynamic process for the actin or microtubules. Of course, it's being regulated by various molecules and machineries. So to make sure they are properly targeted to its destination. So those are the cartoon of the previous growth cone we just showed. Actually, inside is the there's philopodia dilation, there's philopodia withdrawal, and also there's also stabilization of the microtubule along the path. So what other molecules or machineries that can guide the axons to go where it's supposed to go? Actually, as I said, this is a very dynamic process, and this process is controlled by a machinery consists of a lot of structures, a lot of proteins, a lot of factors. So in general, there's attractive molecules and there's repulsive molecules. So basically, those molecules are lining along the path of the axon axon guidance. If it's attractive molecule, the axon will grow towards this chemical attraction, and if it's a repulsive molecule, then the axons will be repelled from the molecule. So here is a cartoon showing the network that's responsible for a retraction and also responsible for extension. In terms of the detail, it might not, it's not that too important for you guys at this point. But one important point you guys need to know is the axons are growing in a very well-regulated way. It's not random. It's not going anywhere. It's not going everywhere, okay? So there are those molecules, and there are all the signaling pathways controlling where the axons are going. So where are the axons are going? The axons are going to search its postsynaptic partners. So in terms of the chemical synapses and in terms of excitatory synapses, the axons are going to look for the dendritic spines. So here is a picture showing the structure of the dendritic spines. Those spines, so this in the middle, this big chunk is called the dendritic shaft, and also those they have the lower part and the upper part. There are two dendritic branches, and along the dendritic shaft, there's tens of dendritic spines, or these protrusions sprouting out of the dendritic shaft. As you can see, there's a spine head, and there's also a spine neck. So this is basically give you an idea how the dendritic spines look like. Again, here is a much more detailed structure of the dendritic spines with the electron microscope, actually with the very high resolution. Again, you can see on the left, there's the dendritic shaft, and also you got a small protrusion, and out of the dendritic shaft labeled with S, that's a spine. And also, if you you can do a reconstruction based on these EM pictures and get out of the structure of the dendritic spines. Indeed, on the right, those are the dendritic spines that are being reconstructed from the EM picture. So in general, the dendritic spines, there's also different types of dendritic spines. And in terms of their morphology, we got a filopodia, which are very long and thin. And also we have the thin spine, we have the stubby spine, we have also the mushroom spine. So in general, the morphology of the dendritic spine is an indication of how mature the spine is. Actually being considered, the filopodia is an immature spine in terms of the thin spine, stubby spine, and mushroom spine are the mature spines. So when the axons find the synapses, they'll form a special structure called synapses. The axons and the spines form the synapses. So here is a structure or a cartoon picture showing the structure of the synapses. So on top part is the axon terminal or presynaptic terminal carrying the synaptic vesicle. There are neurotransmitters in the presynaptic terminal. So the postsynaptic of the synapses is located on a dendritic spine, actually. As we are seeing, so under an EM structure, you identify a synapse, there's a postsynaptic density, right? So here, this cartoon kind of showed you why. Because in the postsynaptic side, there is a variety of skeleton proteins, and there's actin, there's PSD95, and there's mGlua, there's CAMK2. There's a bunch of proteins tethered together, form a slot or form a very dense structure that stabilize those receptors like AMPA receptors or NMDA receptors at the postsynaptic side. So that's why under the EM picture, you saw a very dense structure at the postsynaptic side. It's called PSD. Of course, in and between the presynaptic terminal and the postsynaptic side is a very thin gap called synaptic cleft. As we mentioned or we talk about, the nervous system consists of neurons and glia. So previously we talked about neurons, right? They play a very important role. They communicate with each other. There's a variety of neurons in the brain, and they have different functions. Some are pyramidal, some are inhibitory, and also they have different types. They might carry different functions. So how about glial cells? So in general, the glial cells, there's also a different type of glial cells in the central nervous system. In general, those are the oligodendrocyte. There's also astrocyte and microglia. So here's a cartoon showing a different type of glial cells. You can see they have a different morphology and they play different roles. So here, this slide just tell you a little bit detail about the function of different glial cells. So in terms of astrocyte, they make contact to both the blood vessels or the capillaries and the neurons in the CNS. It can regulate or modulate the ions, chemicals in the extracellular fluid and provide structure support for the synapses. Another type of glia is called oligodendrocyte. So for the oligodendrocyte, we have been talking about axons, right? So another thing I haven't mentioned too much, and will some other professors will give you a detailed description of the communication between different neurons. Actually, they are going through electric signals, right? That's why the neuronal system can respond in a very quick fashion, because it's the synaptic transmission is very fast. So in order for that to happen, imagine if you have a wire. If the wire is naked, right, then it will have leaks, right? The electron cannot be transmitted very efficiently. So for the oligodendrocyte, one of its major rules is to form a myelin sheath that wrap up the axons in a way can isolate the axons, can isolate the wire with the insulator. So in another way, then the synaptic transmission can happen in a very efficient fashion. In this way, there will be no leaks. So that's one of the major rules for the oligodendrocyte. And another major type of glia is called microglia. So you guys might have heard we have a blood-brain barrier, right? In another way, the immune cells in the blood system cannot get into the brain. So then the own immune system inside the brain is a microglia. It serves as a scavenger, or can degrade dead cells and then protect the brain from invading microorganisms, instead of just like a immune cell, like it's the microglia. So this one gives you a sense of, in the lab, how astrocyte really look like in the culture system. So the panel A shows the astrocyte in green, and panel B shows the microglia, and also in red is the dendritic spines. Of course, you'll see the microglia can surround the dendritic spines. And also at the bottom of the diagram, it shows you how the glia cells can actively participate in synaptic function. Actually, from A, B, and C, you can see the glia cells can play roles in synapse formation, in synapse elimination, and also in neurotransmission and plasticity. So for A, you can see the glia cells or astrocyte, they can secrete all this kind of morphogenic factors such as BDNF that can regulate the axonal growth and the synapse formation. And also for the microglia in the middle, it can mediate the dead synapses or the dysfunctional synapses through the phagocytosis in a way. And also the glia cells and astrocytes can also regulate the neurotransmission by engulfing all the neurotransmitter that's released from the presynaptic terminal, which can regulate the synaptic transmission and the neuronal plasticity. Okay, so in terms of the major type of cells in the central nervous system, we'll stop there. And next, we'll move on to introduce the synaptic transmission and the synaptic plasticity. So from the first part of the lecture, I hope you guys can appreciate that in general, the central nervous system consists of neurons and glias, and both neurons and glias have different subtypes, and they develop from the single cell, and also from the neuronal progenitor cells, and from the glial progenitor cells also, and then they develop into different types. They locate into different part or different places of the brain, and they carry out different functions. So first, I hope this will work. I'll show you a brief video showing the synaptic transmission it has voice in there, and I hope you guys can hear it too. If you cannot hear it, let me know. I can just, talk you through it a little bit.
The human brain, one of our most complex organs, allows us to move, feel, think, and learn. Inside the brain, a dynamic network formed by billions of nerve cells processes information via electrical signals. Each neuron receives information from thousands of other neurons via multiple processes called dendrites and transmits information via a single axon.
Professor Zhang?
Yes.
We're able to hear it, but it's quite quiet. Would you be able to turn it up a little bit?
Stored in synaptic vesicles.
Thank you.
These chemical messengers bind and activate postsynaptic receptors, initiating a new electrical signal in the next cell.
Okay, so basically this is just a video. I got it from YouTube. Actually, somebody made it a cartoon based on the current understanding of the synaptic transmission, how it works. This is a very, to me, it's a very fascinating video, and you can get at least get a brief idea of how the transmission, synaptic transmission works. So as we have emphasized along the way, the neurons are not alone, right? They communicate with each other, they connect with each other. So the specialized area or place to communicate or connect with each other is called synapses. So the synapses, we have shown its EM pictures, right? There is those bubblish-like structure in the presynaptic terminal, and also there's PSD on the postsynaptic side. And basically, this is the cartoon showing you the synaptic transmission, which is very similar to the video I have just played. Once the electric signal coming in is called action potential, and somebody else will lecture you guys specifically on that part. When the action potential comes in, when the neurons are excited, it will fire action potential. Okay, when the action potential coming in, it will trigger the release of the neurotransmitters, and then the neurotransmitter will travel across the synaptic cleft and bind to its postsynaptic receptors. And a lot of the postsynaptic receptors are ion channels. They can also be GPCR coupled receptors. Upon the binding of a ligand or its neurotransmitter, they'll be activated, they'll be open, and then leads to the polarization of the postsynaptic neurons, which in turn can fire action potential, and then the signal can be transduced from one neuron to the next, then to the next. In terms of neurotransmitter release, and there is a whole bunch of neurotransmitter release machineries has been worked out by mainly by Tom Stoff and some others. Again, this is awarded the Nobel Prize in 2013. So here's a cartoon showing some of the machinery that's responsible for the neurotransmitter release. You might remember a little bit from the video that when the electric signal comes in, it will trigger the calcium increase in the presynaptic terminal. So the increase of the calcium can trigger the fusion between the synaptic vesicle and the presynaptic membrane. So once the fusion happens, the content or the neurotransmitter that's inside the synaptic vesicle will be released outside of the presynaptic terminal and then into the synaptic cleft. So here are some of the proteins or the machineries that's responsible for that process. So it's good to know for you guys that this is the current model that has been worked out through decades of work in the lab. That's this is the current understanding or the general conception of how the neurotransmitter is being released. So first of all, you might remember a little bit that the vesicles in the EM structure I just showed you in the chemical synapse, they are kind of all over the place, right? So first of all, there is a docking or priming process happens. It's like all these vesicles have to be aligned with the presynaptic terminal and ready for the fusion to happen. Of course, there are multiple proteins ready for the docking and priming. And then once the electric signal or the action potential comes in, the calcium concentration will rise. Then the calcium will activate a calcium sensor in the machinery. The calcium sensor, once it's bound to the calcium, it will lead to conformational changes and will bring the synaptic vesicle to the presynaptic membrane, and then leads to the fusion of the synaptic vesicle and the release of the neurotransmitter. So once the neurotransmitter being released from the presynaptic terminal, where it go? It will travel through the synaptic cleft. I have been telling you, synaptic cleft is a very small space or very small gap between the presynaptic terminal and the postsynaptic neuron. So once the neurotransmitter being released, it will bind to the postsynaptic receptors. So a lot of the postsynaptic receptors are ion channels, and some of them are ligand-gated ion channels. So the a majority of the excitatory neurons in the human brain or in the central nervous system are glutamatergic neurons. In another way, the major presynaptic receptors are glutamate receptors, and it can be further divided into AMPA receptor, NMDA receptor, and QN receptor. So here we are going to focus a little bit on the AMPA receptor and the NMDA receptor, not too much on the QN receptor. Of course, there are some other type of neurons, right? So there are, GABAergic neurons, there are acetylcholine neurons, there are some other neurons that, the presynaptic terminal will release different neurotransmitter and also activate the postsynaptic receptors and finish the neuronal communication or the information exchange between different neurons. So a way of detecting the postsynaptic response is electrophysiology. This is a technique, and also, again, that's also the technique alone was pioneered by Sekman and Irwin. They also got a Nobel Prize for their discovery of this fantastic technique that can detect the electric signal between the synaptic transmission. So this is a way of detecting for using the electrophysiology method to detect AMPA receptor and NMDA receptor response at the postsynaptic side. As you can see, for the AMPA receptor, if it's activated, it has a quick up and then comes back quickly. For the NMDA receptor, as compared to the AMPA receptor, the rise and the comeback is a little bit slower. And then here at the bottom is a combination of the postsynaptic current combined with AMPA receptor and NMDA receptor. And also on the right is just showing a specific for the AMPA receptor and a specific for the NMDA receptor in terms of the APV and the CNQX antagonist that's relatively specific to NMDA receptor and AMPA receptor. So people might ask why NMDA receptors and AMPA receptors have a very different response upon glutamate release from the presynaptic side, since they're both glutamate receptors. So here is the reason why. So in terms of the NMDA receptor activation, it needs two factors to be coincide with each other. Then the NMDA receptors can be activated. First of all, you need glutamate release. And the second of all, you need a depolarization for the postsynaptic neuron. So only both these conditions exist can trigger the activation of the AMD receptors. So that's why there is, upon glutamate release, there is a slight delay for the AMD receptors to activate. Actually, it's waiting for the depolarization of the postsynaptic membrane. Then this AMD receptor can be activated. While for the AMPA receptors, its activation actually only requires the ligand binding or the glutamate binding. It does not require the depolarization. So here is the structure of the AMPA receptors. You can see it's a tetraheteromer. It's a heteromer with 4 different subunits. And upon the binding of the glutamate receptors, AMPA receptors can open the channel, and then the sodium ion can get into the postsynaptic neurons, and then leads to the depolarization of the postsynaptic neuron, which again now fulfill the activation of the AMD receptors. So if you guys can still remember, at the previous slide, I have showed that the AMPA receptor activation is much faster as compared to AMD receptor. So because the AMPA receptor only need the ligand binding, while the AMD receptor need to wait for the AMPA receptor mediated depolarization to happen before it can be activated. I mean, in terms of the details, you guys don't need to remember. So for all these postsynaptic receptors, actually, there's a topology, right? They have an N-terminal, they have a C-terminal. The N-terminal can be outside of the neuron, C-terminal can be inside. There's a topology going on. And also, there are different part of the receptors. Some are responsible for the channel to open, some are responsible for the channel conductance, some are responsible for the channel trafficking, and some are responsible for the channels to be tethered on the membrane, and they all play some different roles in regulating the trafficking or the function of the postsynaptic receptors, which plays important role in regulating synaptic plasticity and learning and memory. So we have been talking about synaptic plasticity for a while. So what is synaptic plasticity? So you might remember that for the pyramidal neurons or the projection neurons, the dendrites has a lot of protrusions, those called spines, right? So those spines can be formed, and those spines can be eliminated. And typically, as I said, so the most excitatory synapses are formed from a axon terminal in a close proximity with dendritic spine. So typically, most spines carry synapses. So if a spine is formed or eliminated, then the synapse can be formed and eliminated along the way. So in a way, the synapse or the spines are dynamic. They're not just static. So based on a certain stimulation or a certain learning experience or a certain some kind of life event, it can trigger the formation or elimination of the spine. This is a very dynamic process. And also, now you know that the synapses, right? They have the presynaptic terminal, and also you have the postsynaptic membrane. So you'll see if there's no formation or no elimination of the spine, there can be also changes at the synaptic level. That's called synaptic efficacy. So in a way, you can think about it. So if something happens to this presynaptic neuron, now the presynaptic neuron release the neurotransmitter more easily, then the communication between the neurons can be enhanced, right? So another way from the postsynaptic side, let's see now, if you have more postsynaptic receptors lining on the postsynaptic membrane, so then upon the release of the presynaptic neurotransmitter, then the chances of binding to the postsynaptic receptor and activating the postsynaptic neuron can also be greatly enhanced, right? So in a way, even without synapse formation, spine formation and elimination, the synaptic efficacy or the transmission between different neurons can also be changed by adding new receptors or increasing the synaptic vesicle release probability. So this whole process is very dynamic. As I said earlier, it is also very associated with a stimulation, a life event, or whatever experience. Then the changes will happen at the synaptic level. This is called synaptic plasticity, and has been shown by a lot of data that synaptic plasticity is very, very important for learning and memory, and also maybe information storage in the brain. So what is synaptic plasticity at the molecular level or at the systems level, right? So how can you monitor or record the synaptic plasticity? So here is again, we are using the electrophysiology recording. Here is a acute hippocampal slice. So there's a stimulation electrode. There's also a recording electrode. So if with no stimulation, if you just record the EPSC amplitude at the baseline, you can see it's relatively stable, right? So if you pair the recording with a high frequency stimulation, what happens is you got a dramatic increase in the EPSC. It's called excitatory postsynaptic current. In the amplitude, there's a dramatic increase. This phenomenon is called long-term potentiation. In another word, upon the high frequency stimulation, the communication or the postsynaptic response get dramatically enhanced or potentiated in a way. It's called LTP. While on the contrary, if you pair the recording with a low frequency stimulation, what happens is there's a dramatic decrease in the EPSC amplitude. In another word, it's called LTD, long-term depression. So in this way, the synaptic communication or the synaptic strengths between those two neurons are decreased. So that's why the synaptic current, there is a decrease. So this is a way of very efficiently or very elegantly demonstrating that the synaptic efficacy can be changed upon a high frequency stimulation or a low frequency stimulation leads to a phenomenon called LTP and LTD. So those two phenomena are widely considered the equivalent of learning and memory, or at least at the molecular level, is the best demonstration. So then how to study synaptic plasticity? So I'm going to talk about a little bit research in the lab from now on. So we have seen, we have used electrophysiology, right? That's a very good way of measuring the synaptic plasticity or the synaptic response in the neuron. So another way you can measure the synaptic change is through imaging. So I have already shown you some immunostaining, some EM picture, and this is also immunostaining confocal picture showing this is the pyramidal neuron you can see, and with very beautiful neuronal processes. And also there's a lot of green dot or green puncta you can see, right? On the right part, there's zoom in to look at it. So what are those green dots actually? Those green dots are the signal for the AMPA receptors. This is one of the one type of glutamate receptors. You'll see the green dot allocation is very different from the magenta color, right? The magenta is just a cell morphology. So in another way, the AMPA receptors is not evenly distributed across the neurons. It's concentrated at some places. Those are the spines or the postsynaptic membrane. So this is why this puncta are here. Those are the AMPA receptors. So people have been working on this, and you might have heard of green fluorescence proteins, right? And also this is also the Nobel Prize in chemistry back in 2008 or something around that. So people have been utilizing those green fluorescence proteins to study their protein or gene of interest. So here, what we are using is a pH-sensitive version of the GFP. It's called SEP. So the unique thing about this version of GFP is pH-sensitive. So, inside the cell, there are different compartmentalization, right? So some compartment has acidic pH, some compartment have a neutral pH. So in the trafficking vesicle, the pH is acidic. So for this specific protein, when it's inside the trafficking vesicle, the fluorescence is quenched. So once the vesicle is fused to the cell membrane, now it's facing the neutral pH outside the cell, then there will be a great increase in the fluorescence intensity. So in such a way, we can specifically measure or monitor the membrane population of the receptors. So what we are trying to do actually, so people through electrophysiological recording and through biochemistry and some other measures, people have studied the postsynaptic receptors. In particular, I'm talking about AMPA receptors in this case, and they have known that AMPA receptors, at least in vitro, is closely related to LTP, LTD, and some play important role for the CNS function. But in terms of how AMPA receptor is correlated with animal behavior or animal in a live animal, it's not very clear. So what we are trying to do here is trying to use a imaging method to monitor the trafficking and function of AMPA receptors, which is one type of glutamate receptors that play important role, and to study how it's correlated with animal behavior. So the type of, first of all, in order to image those AMPA receptors, we have to label AMPA receptors, right? So the method we use is called in utero electroporation. So this is the diagram, how this experiment is performed. Logically, it's relatively simple. You just inject the DNA of interest into the lateral ventricle. You still remember that the neurons are developed from the ventricular dome, right? So, and also it has different stages of development. Let's say at E12, it developed into layer 6, E13 developed into layer 5, and E14 developed into layer 4 or layer 2, 3. So there is developmental timing for those neurons to be born, right? So here we just take advantage of that, and at a particular age, we inject the DNA with interest that's carrying the fluorescent protein fused with our AMPA receptors, and then we give an electrical pulse which will guide the DNA into the neuronal progeny cells. Then once the neuronal progeny cells migrate into its destination, the neurons will carry the GFP fused AMPA receptors, and we will be able to image those fluorescence labeled AMPA receptors and then monitor its dynamics at a certain stimulation, at a certain learning paradigm, to see how does it correlate with animal behavior, how does it correlate with learning and memory. So these are the pictures showing we can transform a decent population of neurons with expression of our fluorescent tagged AMPA receptors. And then what we do is we do a cranial window. You know that the the mouse skull is opaque, right? You cannot image through the mouse skull. So what we are choosing here is do a cranial window, which basically remove a small piece of the skull and then replace it with a cover slip. And then we can image through the window and then look at the dynamics of the fluorescent labeled AMPA receptors inside the brain of a live animal. So here are some examples to show how cranial window is performed. You can see, especially on the right, if you do it with some practice, then you can see there's not much bleeding. We can see very clear pattern of the vasculature, and we can keep this window clean for a relatively long term, and then we'll be able to image through those windows and look at those postsynaptic receptor dynamics. Those are the, as I said in the very beginning, we have different brain structures, right? We have cerebral cortex, we have campus, we have cerebellum, and inside the cerebral cortex, there's also subdivisions. It's called there's sensory cortex, there's motor cortex, there's vision cortex, and there's prefrontal cortex. So again, it's divided into those sub areas based on their function or based on control of animal behavior. So here, what we are trying to do is to use the barrel cortex. The barrel cortex is also called the somatosensory cortex. The reason we choose this because our experiment animal is mice. So for the mice, for the rodents in general, so those guys, they come out and to be active at night, right? So their visual system is not particularly strong. However, the sensory system is very strong. So and also through years of study, people have found that the barrel cortex is the brain area that receives the information from the whisker. So you can see specifically for this barrel cortex, it's very interesting phenomena. Each and every whisker can project to a specific barrel column that's received information from each and every whisker. So there is a very good specificity. Let's see, this is C2, this is C1. So there is a specialized area that's responsible for the information got from the C2 whisker. So on top of that, I'm not going to go into too much detail. So on top of that, so we can also use another method to map out this corresponding region in the brain, and then look at what are changing in terms of AMPA receptors if we do some kind of whisker stimulation. So here is the setup, and this is the animal being anesthetized, and this is with the cranial window, and this is the objective, and then we can see what's changing inside the brain. So here is how can we, what type of images you can get, or what type of images you can image inside the mouse brain. So again, the green is for the receptors we are interested in. The magenta is the morphology marker that can help us to quantify. As you can see, we are imaging from the top of the brain going deep inside. We can see all the neuronal processes, and then it converges into the cell body. So this is the 3D reconstruction from the previous Z-stack. We can see the cell body inside the brain, and then projecting the apical dendrite to the top, to the brain surface. And also we see all those brain puncta. Again, those are the puncta of the AMPA receptors that are located at the top of the brain and form synapses with some other neurons. So what we can do is we can do some kind of whisker manipulation and then ask how or if there are changes of AMPA receptors upon our stimulation. So as shown here in the control group, we don't show much increase or much change in terms of AMPA receptors, while in the stimulated group, we see a dramatic increase in the AMPA receptor content in the dendritic spines. I'm not going to go into too much details into this, and then we move on to next part. So again, what we're talking about, we are really interested in is how does this relate to learning and memory? How does it relate to animal behavior, right? So next, what we are doing is we're trying to train the animal to learn a behavior. In terms of time, I'm not going to do too much detail. So what we are training the animal to learn is, you see, this is a glass pipette, right? So one whisker is poking into the glass pipette. So this animal is not anesthetized. This is a weak being animal. What we are training the animal to learn is upon the whisker deflection, the animal need to poke its tongue out and lick for a water reward. As shown here, upon whisker deflection, the animal can poke its tongue out and lick for the water reward. This is a training paradigm the mouse is supposed to learn. Again, there will be 4 types of behaviors out of this paradigm. So basically is when it's received whisker deflection, the animal is going, then it's called a hit, then can get the water reward. If it doesn't go, it's called a miss. So if it doesn't receive the whisker deflection, it goes, it's called a false alarm. If it doesn't go, it's called a correct rejection. Then based on this 4 type of behavior, we can calculate how well the animal learned task. That was fraction or correct becomes simple. We can see well does the animal learn. Also, how the post learning another is to grab food, and it's called reaching task. And he's and successfully reach out and get it, place it, and he can see. And so having the in the animal using that, so the too much, of course, we are and also so typically and some the floor, so don't have to get some task or to again, you can use learning task to assess how the animal task how this to show how and I'll show you one out of this interested in how the text related written. So
Professor Zhang, are you able to hear me?
yes.
I think your connection is breaking up since when you were explaining.
Again?
Yeah. Maybe can you go back to the go no go paradigm? And yes, I think here is when it started to break up. Could you explain this in the following slides?
Okay, sure. Sorry.
It's okay. Thank you.
So as I have just showed that, we ask the animal to go once it's received a whisker deflection. So there will be 4 behavior outcomes. If the whisker deflection comes, the animal is supposed to go, then can get the water reward. If the animal didn't go, it's called a miss. Then if the deflection didn't come, the animal is supposed to withhold, right? So then it's called a correct rejection. If the animal goes, that means he doesn't know he's supposed to withhold. That's called a false alarm. So based on the paradigm, there will be 4 outcomes, and based on the 4 outcomes, we can calculate how well the animal learned the task. And then we in the whole time, we are able to image AMPA receptors and trying to correlate how does AMPA receptor trafficking or how does AMPA receptor dynamic correlate with this learning, and so get a deep understanding. So next, another brain area we are interested in is the motor cortex. So here we are training the animal to grab a food pellet and then to reach its forearm to grab it. And this is another called forelimb reaching task. As you can see in the video, the animal can grab the food and then eat it. So for those who have housed animals, might know, for you guys might be a little bit out of reach, that in the lab for the animals, they have a mouse cage, right? They have bedding at the bottom, and also they have a grate. Sometimes you have the water coming in, and you have the food. So that's pretty much it. So that's the lab animals. So typically for the animal to get the food, the mice will grab the grate and then bite, right? So then a big pellet will become small and drop to the floor. Then the animal will just hold it and eat it. So it does not have to extend its forearm to reach it. So in another way, so this is a new and novel task for the animal. The animal need to learn to get the task to get the food. And here is another video showing the animal can do the same task under the microscope while head fixed, and we can image in the meantime when it's grabbing the food. As I said earlier, initially we are interested during this forelimb reaching task, what changes are happening in the motor cortex. So interestingly enough, not only we observe some changes in the motor cortex, there's also changes in the visual cortex. So here are some examples. So in another way, I'm not going to great detail that for this task, for this learning behavior, it's not specific to one brain region. It's also there are some other brain regions that's correlated, that's participated in the process. In a way, there's also changes for this forelimb reaching task in the visual cortex in terms of the AMPA receptors. So here is pretty much my last slide. We're going to summarize. I hope during the course you might have appreciated that the human brain or the mammalian brain is an extremely complex organ. It can perform a variety of behavior, and it controls pretty much everything we do and we do not do during our daily life. It's somewhat how to say, it's a very, very, very advanced computer. Let's put it that way. Very, very, very advanced computer. So, I mean, for a computer, you can perform such thing, right? Perform some simple task. But for the human brain, it's kind of similar like computer, but much, much more complex. So you have input. As I said, you can have sensory input, you can have visual input, you can have olfactory input, you can have a lot of input, and all the information will come together. And then there will be processing between different brain structures, between different brain areas. There will be communication between them. And then they come up with a solution. They come up with a response. Of course, that's mostly carried out by motor cortex and the cerebellum to some extent, then you come with an output. So that's how the human brain works, right? So it's and also in terms of how the memory is stored, there, of course, there's some kind of theory. It's not fully approved yet. It's also kind of similar to the computer. So in the computer, you have 0, 1, 0, 1, right? There's certain kind of pattern, then you can encode the information into the pattern. So in the human brain system, now we'll see we have a lot of neurons, right? We have a lot of dendritic spines. We have a lot of synapses, right? So for the synapses, I also talk about synaptic efficacy, right? So it's not, the synapse can be 0 or 1. So 0 is eliminated, it's gone, right? 1 is new formation, it's there. So, and also between 0 and 1, you might be 0.1, 0.2, 0.3, 0.4, 0.5, depends on the synaptic efficacy. So in a way, it's somewhat constructed similar to the computer chips, but it's also much more complicated. I hope through this course, you can appreciate how complex the human brain is. And also, I hope you can have some curiosity, at least to explore, or at least to ask what we don't know about the human brain, what we have known about the human brain, and what else can we know, right? And hope you can get that. So the major take-home message from this lecture, I summarized to be this 3-part. First of all, I want you guys to be curious. Always, always be curious. So if you are not curious, if you are not interested in something, you're not going to work hard. You're not going to dig deep into the problem to get answers. In terms, if you want to do science, if you want to pursue science as your career, always, always be curious. And also second is to get to the bench, okay? So this is particularly important for biomedical research and also for neuroscience as well, because this is a experimental major, right? So whatever idea, whatever theory you have, you need to design experiment to test whether your idea is correct or whether your theory is correct. So you need to get to the bench. So this is very important. And also, thirdly, this is also important if you decide to pursue science as your career, is to be persistent. So there will be a lot of bumps. There'll be a lot of failures, a lot of obstacles going forward. You might form a theory, you might form a hypothesis, you might, have some kind of idea. It could be right. A lot of times it's not right, or it's only partially right. So then in the process, you need to formulate, you need to correct, you need to make changes or make make adjustment. But then you can find out the truth behind it. So in another way, there's a lot of failures along the way. So if you're not persistent, then in the end, you might not get to the truth. Okay? And thank you for your attention. And let's take some questions. Thank you.
Thank you so much for the lecture. I think the way we're going to do this question portion is we're going to ask students, if you have questions, to raise your hand, and then we'll call on you. If you go to the reactions function at the bottom of your screen, you should have the option to do, I guess you can do a thumbs up or a clap is what I see. So you can do either of those.
Yeah, sure. Give me a second. So should I? Yeah, I'll just exit. Let me see, how do I exit? All right, just give me a second. Yeah, just go ahead. Whoever has
Oh, I see. Yes, if you click on participants, you can raise your hand. So, sorry, I might butcher some names.
How can I? Okay, just pause, share, participants. Okay, I got it. Yeah. Any questions so far?
Siddhant Sharma.
So you can just speak out.
So can I just use my microphone and speak out the question?
Yeah, go ahead.
Okay. So will you consider patch clamp electrophysiology as one of the major or the forefront techniques in neuroscience?
Well, I mean, it's tough to say. It's well, I wouldn't I wouldn't call it far front. Okay, so this is the most classic technology in neuroscience. Let's put it that way. So of course, there are caveats for this particular technique, being it's extremely slow put, and if you want to be a good electrophysiologist, you have to be very, very patient, because patch clamp needs a lot of practice to be good at it, and you can only patch one cell at a time. Now there are techniques saying, okay, we can patch 8, we can patch 12, but still, as compared to some other method, this is a relatively low throughput. This is still the golden standard of detecting the neuron activity or the neuron response in terms of accuracy, and this is also a very fast. And people have tried to use voltage-sensitive dyes to replace electrophysiology, but so far has not been very successful yet. It can work to some extent, but in terms of accuracy, in terms of sensitivity, it cannot compare to electrophysiology or patch clamp yet. And this is definitely a very, very unique, a very, very useful technology. Yes.
Thank you for answering my question, and that was a really good lecture, Professor.
Thank you.
Janice?
Hi. I was a bit confused on the categorizing neurons. Can you explain how morphology works in like categorizing what kind of neuron it is? And like, I was a bit confused on how like different morphologies might affect the function of the neuron.
Okay. So let's see. Just give you a broad sense, okay? First of all, we can divide the neurons into exciter neurons and inhibitor neurons, right? So the morphology of the exciter neurons and the inhibitor neurons are vastly different. The inhibitor neurons don't have much spines. Of course, they have a lot of neuronal processes. For the exciter neurons, they have a lot of dendritic spines. All these spines can form synapses with another neuron. For the inhibitor neurons, it doesn't mean they do not form synapses. So the inhibitor neurons, I didn't go into too much detail. The inhibitor neurons can form synapses onto the dendritic shaft, not to the dendritic spines. So in terms of how the neuronal morphology affect the neuronal function, let's put it that way. So the neurons actually carry their function in the end point of the neuron carrying function are the proteins, right? You have a lot of proteins, a lot of genes express proteins and carry the function. Let's put that way. So if you have a dendritic spine, right, that's stubby. And also if you have a dendritic spine, that's filopodia, that's long and thin. So in terms of transporting proteins in and out of the spine, it's going to be different. So when we talk about neuron, depends on the neuronal shape. It can express different patterns of proteins. It can express different type of neuron transmitters, and also it can express different type of postsynaptic receptors that will dramatically affect the function of those neurons.
I think a helpful thing, could you distinguish between excitatory and inhibitory?
What do you mean by distinguish? So those are defined by their function. So the excitatory neurons, when the excitatory neurons are activated, right, it will send a signal to the next neuron for the neuron to be excited. When the inhibitory neurons are excited, the inhibitory neurons will send a signal to its next neuron, stay quiet, don't get excited. Is that clear?
Yeah, I think that is helpful for the previous question. Okay, Shikang.
Oh, hey. Oh, thanks. I have a question that one of your slides have shown the two different response curve of two receptors.
Mm-hmm.
Yes, and in this slide, the AMDA receptor seems to have a more spiking-like response curve. And my question is that they have two different receptors have two different response curve. So what will these two different response curve lead from the functional perspective?
Okay, so that's a good point. So we're talking about those AMPA receptors and NMDA receptors, right? So
right.
Functionally, actually, so I'll go to a little bit in detail. Hopefully, don't confuse you too much. So those two receptors are both glutamate receptors, right? In other words, those two receptors go hand in hand. Actually, you will find neurons have both AMPA receptors and NMDA receptors, except actually during development, there is a phenomenon called silent synapses. So what does it mean? The silent synapses is morphologically the synapse is there, but the silent synapse means when a presynaptic signal release the neurotransmitter, in this way it's a glutamate, the postsynaptic neuron does not give a response. That's why it's called silent synapses, because the postsynaptic neuron cannot respond to a neurotransmitter release. So what is on the silent synapses? The silent synapses have NMDA receptors, but does not have AMPA receptors yet. So that's a development process. There will be synapse maturation. Later on, AMPA receptors will come to the silent synapses. Then the silent synapses can be responsive to the presynaptic release. So that comes to the curve. So because the AMPA receptors, as I said, respond to the glutamate release, that's the only condition that AMPA receptor require to be activated. If it's glutamate there, the AMPA receptor can be activated. While for the NMD receptors, if without AMPA receptors, the NMD receptor is there, the glutamate can bind AMPA receptors, but because there's no AMPA receptors, which can lead to the postsynaptic cell depolarization, the AMPA receptors cannot respond. So in terms of function, so for the AMPA receptors, you might remember, you might not. So there is called tetraheteromer, right? So if you go to detail, there's called glutamate receptor AMPA type, GLU A1, A2, A3, A4. And also there's combinations. And for AMD receptors, also there's combinations. It's a heteromer, right? So they have different subunits. For the AMPA receptors, it's mainly a sodium channel. For the AMD receptors, it can also permeate calcium. So in terms of the function, you can see the AMPA receptors, basically AMPA receptor opening can lead to the sodium influx into the cell, while the AMD receptor opening not only, well, it does not, because the AMPA receptor is, responds to the glutamate release earlier, it also, leads to the calcium, the sodium influx earlier. So the AMD receptor, the major response for the AMD receptor is actually for calcium to entry. It's more like a signaling receptor in a way. For the AMPA receptor, it can depolarize the postsynaptic cell. I think for some later lecture based on the course arrangement, there will be people talking about the communication, which is the action potential. How does the action potential generated? There will be sodium entering, and then the potassium will do go out. I'm not going to go into too much detail. In terms of function, you can consider this. So mainly the AMPA receptor is responsible for the sodium entry. For the NMDA receptor is responsible for the calcium entry.
Yeah, and could I think that the AMPA's role is the AMPA's role is to give initial response and for the later sequence events.
Yes,
the
in a way.
Okay.
So there is a statement that the AMPA receptors mediate the majority of the fast synaptic transmission in the central nervous system. All the fast synaptic transmission are mediated by the AMPA receptors. So we can, based on the curve, because it's responsible faster than the NMDA receptors.
All right, I get it. Thanks.
Okay, all right.
Eza, is that how you pronounce your name?
Yeah, that's it. Concerning the SCP, what's the relation to it, the SCP and the membrane pH? How does it affect the vesicle? And also for the excitatory postsynaptic potential, I want to know how it regulates frequency stimulation with the LTP and the LDP, because I didn't really get the synaptic plasticity for those ones.
Okay, so for SEP, it's basically a variant for the GFP protein. So one of the Nobel laureates for the GFP discovery is Roger Chen, who was at the UCSD. Unfortunately, he died prematurely a couple of years ago. So what he did is actually, so initially when the GFP was discovered, it's fluorescence, right? You get a green light after GFP protein from the jellyfish, but it's not bright enough to be used as a tool in the biomedical research. It's Roger Chen who did a lot of construction or reconstruction, if you call it, would like to call it, to make the GFP much brighter. It's called eGFP. Later on, it's called eGFP. It's called enhanced GFP. So in terms of the SEP, it's one of the variant out of the screening to make the GFP better. So they find this one is just a couple of mutations in the amino acid sequence make this particular GFP variant sensitive to the pH. So when the trafficking vesicle budding off the ER, right, the rough ER to traffic the protein content or whatever content into the membrane, the lysosome, right? Lysosome is an organelle inside the cell. So inside the lysosome, it's very acidic, right? So inside the trafficking vesicle is also acidic. It's about pH 5.2-ish. So under that pH, the SEP fluorescence is quenched. Once it's trafficked to the membrane, it's fused to the membrane. Now it's facing like the pH outside the cell. It's about 7.2 or 7.4. It's neutral. So now the fluorescence intensity is dramatically increased.
Okay. Okay, so what about the excitatory postsynaptic potential and how it regulates frequency stimulation in order to either make an LTP or LDP?
Yeah, well, I mean, it's it's the other way around actually. So it's the depends on the frequency of the stimulation. The postsynaptic cell, okay, will have a response if you have a high frequency stimulation, or in another word, in a simple way, if you have a strong stimulation in the presynaptic side, right? Your trigger release, right? So then what happens is the postsynaptic cell will give you a better response.
All right.
Of course, this stimulation is repeated, right? It's not like one time, it's repeated. So if you do it multiple times, will cause this phenomena called LTP, right? So what this phenomena does in a simple way you can consider is the stimulation, the high frequency stimulation leads to a strengthening of the connection between those two neurons.
Okay. All right. Then
or
if it's
for
low,
the LTD, for the LTD or for the low frequency stimulation. So you can try to think it this way, but it's not exactly accurate. So, but this can help you understand the thing. So if you're being scratched, right? If you're being scratched, like at the similar force, 5 or 10 times, you might get desensitized, right? You don't feel the scratch too much anymore, right?
Is it
So
like
this
something like summation? Sorry for interrupting. Is it like summation of impulses, kind of? Should we say
in a way,
like
you
that?
can LTD might is called similar to a desensitization. So if you keep on if you would consider a sub-threshold stimulation, right? If you stimulate, but a sub-threshold doesn't really generate much at the postsynaptic side, then the neurons will get somehow desensitized. If you give a normal, after this training, after this low frequency stimulation, if now you give a normal stimulation, which typically will generate a postsynaptic response, now it won't respond anymore. Then it's somehow desensitized a little bit.
All right, thank you very much.
Okay.
Okay, Kyle.
Kyle, yeah.
Hi, Dr. Zhang. Thank you once again for your
no problem.
Regarding the neurogenesis section for the phase of cell migration, I just had a quick question regarding could you elaborate why you mentioned it being inside out from layer 6 to layer 1, then being then vice versa?
Well, I mean, that's how to say, that's what people have found in the cerebral cortex. That's the way how it's developed. In terms of why is this, we don't know. That's just what people have found using those labeling experiments. We find, okay, the layer 6 cells are formed firstly, and then layer 5, layer 4. In terms of, this is not general to each and every brain area. In the hypothalamus, actually, it's outside in.
Oh, okay.
So, and also in the retina, it's kind of mixed. So, I mean, the whole point is the migration or the neurogenesis, there's certain kind of pattern. It's not random. It's not like, okay, you're born, then you go to layer 1. The next born, you might go to layer 6. It's not like that. There's certain kind of pattern. But instead, in terms of pattern, different brain areas, different brain structures can vary. But there's a pattern in there. In terms of why, we don't, at least I don't know exactly why. Just people have demonstrated it is the way it is.
Thank you.
Okay.
Christy Tam.
Hi, hello.
Yes.
Hi, yes. Doctor, hello. So you spoke a bit on synaptic efficacy, but could you tell us a bit more about how you quantify that? How do you know how strong or how weak the efficacy is, or is it just like a general term?
Good question. It's not a general term, okay? So because how to say, I have been seeing that the synapses is not too big a structure, right? It's a teeny tiny structure, but it also contains multiple nanodomains. Let's say we have a synapse, right? A synapse might have 50 or 100 postsynaptic receptors. Those 50 or 100 postsynaptic receptors, again, is not evenly distributed across the postsynaptic membrane. There are certain clusters, right? So, and also when we talk about the presynaptic release, the fusion between the synaptic vesicle and the presynaptic membrane, right? There is an active zone, but again, it is a zone. It's not an active site. So what do I mean by that? Every time when there's a fusion, that could be at position 1, or could be at position 2, or could be at position 3, or could be at position 1, 2, 3 all together. So that's why, so when we do this kind of electrical recording, there's called miniature release or evoked release. So for this kind of miniature release, or you can call it spontaneous. For this spontaneous release, it could be quantal or subquantal, depends on how you call it. So let's see, for a particular synaptic vesicle, it contains, let's see, 100 whatever neurotransmitter in there, right? So if it released at position 1, it might have a different effect than release at position 3, because it's not exactly the same place. And also take into account of the different postsynaptic receptor cluster. So there could be a neural cluster right under position 1, but a little bit distant away from position 3, you understand? So if that's the case, release in position 1 and position 3 to the presynaptic side, there's no difference, right? This is a release, either 1 or 3. But in terms of a response, that could be different, because the local concentration for the postsynaptic cluster, for the postsynaptic receptor cluster, could be different. So in that way, a release could trigger, at position 1 might trigger a much bigger response as compared to position 3. So that's called synaptic efficacy. So we haven't talked to, so, okay, there could be ways, there could be regulation that the presynaptic release can be more frequent, can release more, right? So then in that way, the communication between the presynaptic terminal and the postsynaptic neuron can be better than less frequent release, right? And also, if there are more clusters of the postsynaptic receptor, in that way, you can think, if you release position 1 or release at position 3, it can all generate postsynaptic response. In that way, if you have more postsynaptic receptors, the synaptic efficacy will be better than you have less postsynaptic receptor clusters.
Oh, okay, thank you.
Suman.
Hello, that was an amazing lecture.
Thank you.
Yeah, the question I had was raised during your lecture by one of the students, and the question is that the inhibitory neurons do not have spines. So, what is the significance of there not being spines in the inhibitory neurons versus there being spines in the excitatory neurons?
How to say, there's some kind of theory actually. So, in another word, there are some parentheses in that slide that the excitatory neurons, and also called pyramidal neurons, and also called projection neurons, right? So they project to somewhere else. So for the inhibitory neurons, it's different. So they don't project. For the inhibitory neurons, it's kind of how to say, called a tiling in a way. So for the inhibitory neurons, sometimes it's just, so I control this area, another neuron control that area. So, but for the excitatory neurons, the cell body is at layer 3, layer 2, 3, or layer 4. It's projecting to the brain surface. So the actual synapses is formed actually at the top layer, it's layer 1. So in another word, so the exciter neurons is really form a circuitry. They talk to each other very, very closely, very, very frequently. In terms of inhibitor neuron, you can think of it's more like a modulatory rule. In terms of why they don't have spines, that's just morphologically, that's the way it is. But it does not mean it does not form synapses, as I said earlier. The inhibitor neurons tends to form synapse at the dendritic shaft, but not at the spines.
Thank you, sir.
Ibrahim?
Hi, so I wanted to ask, so in the wake of an autonomic response, which of the receptors or AMPA, I'm guessing in this case, is going to be responding the earliest as it has the lowest time, or all the receptors in the neuron are going to be equally taking part in that process?
Depends. So we have focused on AMPA receptors, right? So that's one type of neurons. That's called glutamatergic neurons, right? Because this neurotransmitters it uses is glutamate. So there's dopamine neurons, there's serotonin neurons, there's 5-HT neurons. So for all this, the responding receptor will be dopamine receptor, acetylcholine receptor, or 5-HT receptor. So there is a relatively specific receptor responding to the neurotransmitter. It's not only AMPA receptors. Here we are just using glutamatergic receptor, glutamatergic neuron as an example to give you the point.
Roger, thank you.
Are there any more questions? Okay, I don't see any more hands. If everyone taking the course could stay after for like 2 minutes while I make an announcement. But yeah, that's it. So thank you so much, Professor Zhang.
All right, then. Thank you. Hope you guys enjoyed, and hope you guys will be interested in neuroscience. And all right. Thank you.
Have a great day.